Hi everyone, today I am back with some more discussion regarding CSIR NET Life Science Unit 1 important previous year questions with their solution. Today's topic, questions will be discussed from amino acids, which is a very important topic from CSIR point of view. As we all know, amino acids, they have a central carbon alpha, surrounding it is an amino group and an alpha carboxyl group, and there is a variable R group which can have many different functional groups in it. And there is hydrogen. There are different classifications of amino acid based on the R group, whether the R group will be a hydrophobic functional group that is containing CC and CH bonds, or it will be some polar molecule which can interact with water. So depending upon the pH, the charge of the functional groups will be changing, and that is where the charge of the amino acid will also keep on changing based on the pH. Here, some important concepts like pKa, Pi will be used, and questions are also based regarding that. There is a connection with Unit 13 where ion exchange chromatography, generally questions will be linked with the proteins and what charges will be there and how it will be eluted out in chromatography. Let's dig into the details. The first question is from CSIR paper December 2016, and almost every time there will be some question regarding amino acids. Here they are asking, choose the most appropriate pH at which net charge is 0 for the molecule from the data shown below. We can see there are some forms of the amino acid which are given, and as I said, depending on the pH, the forms are keeping on changing. pK is a point at which a functional group will dissociate. For example, the carboxyl group of alpha carboxyl group of amino acid has a pK point of 2.3, which means that when the pH comes to 2.3, this group will dissociate or lose a proton. Okay, will lose a proton. Similarly, amino group, alpha amino group, it has a pK of 9.6. That means when the pH will be coming to 9.6, that time the amino group will lose the proton. After losing the proton, amino group will become NH2, and before losing the proton, it will stay as NH3+. Whereas carboxyl group, after losing proton, it will become CO-, whereas before losing it will remain as COH. So basically, when you have pK point of a group, a pH below the pK point, the group will be present in a protonated form, whereas a pH above the pK point, the group will be present in a deprotonated form. Okay. Now here, when we start with 0 pH, obviously as the pH increases, the different pK points will be reached. So starting from 0, the first pK point will be 2, which is for the alpha carboxylic group. That is why you can see that in the next form, the alpha carboxyl has been dissociated. Okay. Then the next pK point will be there, which is 3.8, which is for the R carboxylic group. Even though both are carboxylic group, R carboxylic and alpha carboxylic group, they will be having different pK points. Okay, it is because of the spacing of the functional group in the molecule. Since the alpha carboxylic group is placed next to amino group, that is why its dissociation will happen at a lower pK value. Okay, so once this is also pK is reached, then this one will also be dissociated. Okay, that is why this form will be different. Then after this pK point, that is the amino group's pK point, even amino group will be dissociated. Now the pH at which the amino acid will have 0 charge, that is what is called as the Pi point or isoelectric point, right? Now we have to calculate the different charges present on the amino acid. Like this, if we don't have anything written on the superscript, then we'll assume it is 0. Okay. So that means this doesn't have any charge on it. So this is 0. This has a +1 charge on it. This also has a 0 charge on it. Okay. So net charge we have to calculate for this form is +1. Okay. Then for this it is +1. For this it is -1. For this it is 0. Now this gets cancelled, and this entire form has a net charge of 0. Okay. This is -1. This is +1, and this is again -1. This gets cancelled, and this entire form has -1. Like this entire form will be having -2. So they are asking at what pH you will find 0 charge. So zero charge we are finding in between this pH and in between this pH, isn't it? Because before this pH it was +1 charge and after this pH it became -1 charge. So in between these two pH only zero charge will exist. That means out of this it should be more than 2 and it should be less than 3.8. So 2.91 is the only answer. Basically you have to find the average. To find out the PI value, you have to find the average. That is 2.02 plus 3.8 divided by 2. So answer option here will be answer 2. Okay. So this is regarding the different protonation and deprotonation. Just now what I said, that groups, if they have the proton, they are protonated. If they lose the proton, then they will be deprotonated. Okay. Then there was a question in June 2021 regarding the pKs of the ionizable group in lysine. Right? So in lysine we have 3 functional groups. One is the alpha carboxylic group. If I write the C alpha, so you will have the alpha carboxylic group, then you have the alpha amino group. I am writing them as protonated, and then you have H, and then you have C beta, you have C gamma, C delta, C epsilon, and to the C epsilon there is one more amino group connected. So here these are the ionizable groups. The question is to calculate the PI of lysine, right? Now what are the steps to be followed here? Each one of them have their own pK point. Alpha carboxylic group has 2.16 pK point. pK for the alpha amino group is 9.06 and for the R amino group it is 10.54. Okay. So all these pK points, now you have to find out what is the PI of lysine, right? So how will you calculate that? First of all, you have to see where the lysine will exist as zero charge, right? Now if I have this lysine, if you see, this is the lysine first form. First form means when you start from 0 pH. Okay. Now this lysine, it has all the functional groups, they have the proton. That means all are in their protonated form, right? Now the first pK point that it will reach is around 2.3. After which the alpha carboxylic group is getting dissociated, isn't it? And the rest of the groups are as it is. Then it will be the next functional group pK, that is 9.6, after which the alpha amino group gets dissociated. Okay. The rest of it is still in the protonated form. Anyways, this was already deprotonated before. After this, there was this 10.5, which is the pK point of the R group. So even this got dissociated now. If this is dissociated now, all are in the deprotonated form. Like that, you are getting total 4 forms. Okay. So for basic amino acids such as lysine, how will you calculate the isoelectric point? First of all, you find out the charge on them. Same way how we did in the previous question. This is 0, this is +1 and this is +1. So total will be +2. Like that, this will be +1, this will be 0 and this will be -1. And what is the isoelectric point? The pH at which the amino acid has a net 0 charge. So this is the form where the amino acid lysine has 0 charge. So you have to find out this 0 charge form. It is present between which two pH points? So it is present between this pH point, which is 9.6, and between this pH point, which is 10.5. Basically this is pKr. pKr means pK of the R functional group, and this is pK2. pK2 means the pK point of the amino group. Okay. So for all the basic amino acids, how will you calculate the PI? By taking an average of the pK2 and the pKr. pK2 is for alpha amino group, pKr is for epsilon amino group. In case of lysine, for other groups you just take the other R group. So in this question they have given the two pK values, 9.6 plus 10.54 divided by 2. That's why the answer is 9.8. In 2021 there was another question to calculate the net charge of the entire peptide, okay, at pH 5. So here I will tell you the question. In December 2023 also there was a similar question. I will be discussing with that also, but I just wanted to show you that this is an important topic and that is why this question keeps on coming time to time. Okay, so let me explain you the December 2023 question, and later you can come back and try to solve this question on your own. Okay, Similarly, here one more question was there in the same exam. The pK of the ionizable groups in the tripeptide below are shown. So here is the entire peptide, okay, and as you know, in peptide amino acids, they are joined by the peptide bonds. So here we have the two peptide bonds connecting the three amino acids, and the different ionizable groups are already marked. So here we have our terminal amino group, terminal carboxyl group, and the side chain of only the central amino acid is ionizable. This amino acid is not ionizable, side chain. Even this amino acid is not ionizable. So while calculating the charge of the peptide or the PI point or anything, you don't have to consider all this. Okay, so they are asking what is the PI for this peptide. Now, how will you calculate over here this thing? So since the peptide has 3 pK points, just like I had told you in lysine also, that means it will have total 4 forms. One will be fully protonated. Fully protonated where it's alpha amino group, alpha carboxyl group, that is the terminal groups, and the side chain which is ionized, they will be all in the protonated forms. So you will have to consider their protonated forms. That means they will be +1, and our carboxylic group, if it is protonated, it will be 0. So total it will be +2. Similarly, after form 1 will be dissociation after the carboxyl group at pK. So you have to dissociate it and write it as CO minus. Basically, you have to draw this peptide. You have to draw this peptide like how we had drawn all the 4 forms of lysine. Like this, you have to draw all the 4 forms. First, you have to dissociate this group. Then you have to dissociate this group. Then you have to dissociate this group. Okay. And same way how we calculated the PI, you have to see where this entire peptide exists in 0 charge. Okay. So wherever it exists in 0 charge, you have to see the pH points before and after that, and you have to take the average. So in all the 4 forms that we have got, PI point will be the average of the pK values in between which the polypeptide chain carries no net charge. That is form 2. But it should be lying between pK2 and pKr. Okay. So therefore, we have to take 9.8 and 10.5. 10.515 is the correct answer. I hope you followed that. Now this is the recent most question which I wanted to show you. It's a part C question from 2023 December exam. The amino acid sequence of tetrapeptides are given below. So here is one peptide, another peptide and another peptide. So you have to calculate, there are 2 parts of this question. First you have to calculate the net charge on the above tetrapeptide. Okay, and second part is if a mixture of the above tetrapeptide is separated on a cation exchange column, then which tetrapeptide will elute last? So let me tell you how to calculate the charge for each one of them. Okay. So whenever you have one particular peptide, let's say you have aspartate and then you have glycine and then you have aspartate and then you have serine. The first thing which you have to do when it is a peptide, you have to note down the terminal amino group and terminal carboxyl group because they also are ionizable groups. Okay. And then you must know which amino acid functional groups are ionizable. So in Leninger you can find this list. For example, aspartate is ionizable, which has a carboxylic group. Glycine is not ionizable because its side chain is just H. Again, aspartate, it will have another carboxyl group. Serine has a OH group, but that OH is not ionizable in 0 to 14 range. So that is why here in this peptide you have 4 functional groups. Now the next step is to write down the pK points of these functional groups. Alpha amino group pK point is 9.6. You can circle it. Then aspartate carboxyl group is 3.7. Okay. Similarly, this is also 3.7. And alpha carboxyl group is 2.3. Right. Now they have asked you to calculate the net charge at pH 7. So you see that these pK points, whether the desired pH is more than that or less than that. I already told you if the pH is less than a pK point, then the group will be in the protonated form. And if the pH is more than the pK point, then the group will be in deprotonated form. So accordingly, you have to write that if pH 7 is less than 9.6, that means the group will be in the protonated form. Okay. If the pH is above the pK point of 3.7, that means group will be in deprotonated form. Right. Same over here also deprotonated. Okay. Same over here also deprotonated. Now what will be the protonated form of amino group? It will be NH3 plus. Deprotonated form of carboxyl group will be CO minus. Deprotonated form of carboxyl group here also is CO minus, and here also it is CO minus. So all this will be -1, -1, -1, and this will be +1. So +1, -1 got cut, and this charge of the peptide at pH 7 will be -2. Okay, so like this you have to calculate the other two also, as I have already shown over here. So calculation of charge at pH 7, as we saw for P is -2. Similarly, if you calculate for Q, it will be +1, and if you calculate for R, it will come to +2. Okay. So here, in case of Q, we will require arginine side chain pKr, which is 12.5, and in R, you will need this lysine. So lysine, anyways, we saw it is having the pKr of 10.5, isn't it? Now coming to the cation exchange chromatography. So what do we know about ion exchange chromatography? If we have cation exchange, that means it will bind with the cations or the positive charge ions, isn't it? That means the resin or the column itself, it will be negative charged. Okay, so this is cation exchange. So column, you can see they are negative charged, and you can see that the positive groups have bonded to the negative charge, isn't it? So out of P, Q and R, so what are the charges on P, Q and R? So on P it is -2, okay, on Q it is +1, and on R it is +2. So I will just write this. So it is -2 and +1 and +2, isn't it? So we know that in cation exchange, the positive groups will be attached. That means when you run P, Q and R through this column, the Q and R being positive charged at pH 7, they will get attached to this column. Whereas P, it is negative charge, it won't be able to bind to the negative charged column. That is why it will elute out. So P will be the first one to be eluted out first, and Q having +1, it will be, if you add the if you start the elution of the bonded cations also, then Q is having less charge than R. So Q will be eluting out next, and R having maximum charge, it will be eluting out last. Okay, so R carrying more positive charge would elute out last. So that's why the correct answer over here is option 1. Yeah, so the correct answer over here, as you can see, this option 1 is the correct answer. Okay, I hope you understood how much how questions come related with other concepts in other units also. That is why it is essential to study related topics together like this. Okay, so that's all for amino acid topic. Let me know in the comment section if you want me to discuss proteins with you and the important topics which has come from this topic in CSIR NET exam. Thank you everyone.